Hello everyone, I am Jayesh Patil from final year B Farm, studying in Shivajirao S. Jonde College of Pharmacy, Asangaon, along with my coordinators Deepak Rathod, Akran Syed and Purva Patil. Today we are discuss about the extraction of silver nanoparticles from leaf extract with the guidance of Mrs. Swati Vaksawar ma'am, Assistant Professor of our college and Department of Pharmacognosy. Let's see. Aim to study the extraction of nanoparticles from leaf extract. Objectives: the extraction of nanoparticles from leaf extract of Clitoria ternatea, preparation of leaf extract, preparation of silver nitrate AgNO3, biosynthesis of silver nanoparticles through bioreduction process, extraction of leaf extract, analysis of silver nanoparticles. Introduction of nanotechnology. Nanotechnology is the fastest growing area of manufacturing in the world today, and there is a increasingly frantic search for new nanomaterials and method to make them. It has been well known that living cells are the best example of machines that operate at the nano level and perform a number of jobs ranging from generation of energy to extraction of targeted materials at very high efficiency. Silver nanoparticles are widely used for its unique properties in catalysis, chemical sensing, biosensing, photonics, electronics, and pharmaceuticals. Silver nanoparticles have a great potential for use in biological, including antimicrobial activity. Antimicrobial capability of silver nanoparticles allows them to be suitably employed in numerous household products such as textile, food storage containers, home appliances, and medical devices. Silver is an effective antimicrobial agent which exhibit low toxicity. The most important application of silver and silver nanoparticles is in medical industry such as topical ointment to prevent infection against burn and open wound. Plants provide a better platform for nanoparticles synthesis as they are free from toxic chemical and provide natural capping agent. Moreover, use of plant extract also reduce the cost of microorganisms, isolation and culture media, enhancing the cost competitive feasibility over nanoparticles synthesis by microorganisms. This is the picture of Clitoria ternatea plant. Synonyms: Bengali Aparajita, English butterfly pea, blue pea vine, mussel, shell climber, pigeon wings. In Sanskrit, Sanskaya pushpi, Aparajita, Saukarnika, Ardrakarni, Girikarnika, Supushpi, Mohanasini, Visha गोगासगिनी, श्वतानमा, विष्णुकर्णदा, अश्वकुरा, इन हिंदी कोयाला, तेलुगु डिनाटिना, मलयालम संगुपुष्पमा, कन्नडा नागरहेडी, मराठी गोकर्णा, पोर्तुगीज फ्लोराक्युआ. Are the picture of leaves of plant Clitoria ternatea. Biological source: it is a perennial herbaceous plant also known as blue pigeon wing. Morphology: drug generally occurs in the form of leaves and leaflets, rhizomes broken with or without intracleates. It is perennial twigs, herbs having 7 leaflets which are elliptic, and obtuse leaves are pinnate 5 to 9 foliate. Flowers are show blue or white, petals are unique style beard below the stigma. Fruit pods are linear and compressed. The pods are 5 to 7 cm long, flat with 6 to 10 seeds in each pod. Seeds are 6 to 10 and black in color. Plant flowers in rainy seasons and fruit in winter. Clitoria purpurea has dark blue colored papillonous flowers, and Clitoria ternatea has creamy white colored flowers which are solitary and very attractive.
Clitoria ternatea leaf. Clitoria ternatea habit is a twining shrub with alternate imperipinate leaves. Above their flowers are solitary or in pairs. The classification of Clitoria ternatea is given in the following. Their family is a Fabaceae, subfamily is a Faboidea, genus is a Clitoria, species is a Ternatea, botanical name is Clitoria ternatea, and common name is Shankapushpa. Chemical constituents. Roots contains taraxerol and taraxerone, pentacyclic triterpenoids and flavonol glycosides, proteins and fatty acids. Seeds contain para-hydroxycinnamic acid, beta-sitosterol, gamma-sitosterol adenosine, flavonol 3 glycosides, ethyl alpha-D galactopyranoside, 3574-tetrahydroxyflavone, 3 aminoglycosides, hexacosanol, and an anthocyanin glycoside. Flowers contain camphophorol and its derivatives, quercetin and its derivatives, and myricetin 3 glycoside. Leaves contains beta-sitosterol, flavonoids, lactones, aparaxitin and clitorin, essential oil, coloring matter and mucilage. Uses. The Clitoria ternatea plant is used as an antihelmintic, local anesthetic, anti-inflammatory, antipyretic, and analgesic, antioxidant, anti-diabetic, anti-ulcer, etc. Silver nanoparticles. Silver nanoparticles have stability to heat and other properties like antimicrobial, antioxidant, antiviral, anti-cancer, and anti-inflammatory activities. The synthesis of silver nanoparticles is a two-step process. The first step involves silver ions reducing to Ag, and the next step includes the agglomeration and stabilization that form the oligomeric clusters of colloidal silver nanoparticles. The reduction steps happens in the presence of biological catalysts. Application of silver nanoparticles. Scientific applications. Due to the surface plasmon resonance and surface enhanced Raman scattering properties of silver nanoparticles, they have many applications such as sensing applications including detection of DNA sequences, laser desorption or ionization, mass spectrometry of peptides, colorimetric sensors for histidine, enhanced IR absorption spectroscopy, biolabeling and optical imaging of cancer, biosensors for detection of herbicides, and glucose sensors for medical diagnostics. Industrial applications. The silver nanomaterials and silver nanocomposites are used as a catalyst in many such as CO oxidations, benzene oxidation to phenol, photodegradation of gaseous acetaldehyde, and the reduction of the para-nitrophenol to para-aminophenol, to catalyze the reduction of dyes by sodium borohydride. Silver nanoparticles immobilized on silica spheres, etc. Methodology. The process followed in methodology is: the first step is literature search, on basis of which the suitable plant is selected. The second step is selection of plant, which is overcase is Clitoria ternatea. Thus, for the experiment, we need to procedure the sample plant, that is the next step, called as collection and procurement of the plant. This step is followed by preparing leaf extract from the collected plant material and proceed by the preparation of silver nitrate. Synthesis of silver nanoparticles by leaf extract is carried out, and then the thus result obtained henceforth is concluded eventually. Procedure. Plant material. Clitoria ternatea habit is a twining shrub with alternate imperipinate leaves. Above their flowers are solitary or in pairs. The classification of Clitoria ternatea is given in the following. The family is a Fabaceae, subfamily is a Faboidea, Genus is a Clitoria. Species is a Ternatea. Botanical name is Clitoria ternatea. Common name is Shankupushpa.
Collection and preparation of plant materials. Fresh leaves of Clitoria ternatea, free from diseases, were collected from Pathare Nursery Home at Kalyan West. Wash them thoroughly 2 to 3 times with tap water and once with sterile water. 20 grams of fresh leaves was finely chopped and added to 100 ml of distilled water and stilled at 60 degrees Celsius for 1 hour. After boiling, it is filtered with the Wortman paper number 1. Filtrate was collected in a conical flask. The collected solution was named as A. Leaf extract of the plant Clitoria ternatea. Next is the solution names. A. Leaf extract solution of Clitoria ternatea plant. Its color is dark color, yellow. And B. Silver nitrate AgNO3 solution. Its color is white. And C. Silver nanoparticles solution. Its color is red. Synthesis of silver nanoparticle. Aqueous amount of solution of silver nitrate was prepared and used for the synthesis of silver nanoparticles. 5 ml of leaf extract of Clitoria ternatea was added to 45 ml of 0.1 molar AgNO3 solution. Then the bioreduction process was carried out at room temperature. Test and characterizations. The Clitoria ternatea were subjected to different phytochemical analysis. Test for alkaloids. It consists of Mayer's test and Dragendorf's test. In Mayer test, 250 ml of crude extract, 250 ml of 1% HCl in water and 6 drops of Mayer reagent was added. An organic precipitate indicated the presence of alkaloids in the sample. Test for tannins, in which one is lead acetate test. In this test, 250 ml of crude extract and few drops of 1% lead acetate solution in water was added. Appearance of white precipitate indicated the presence of tannins in the sample. Test for steroids. 250 ml of crude extract and 250 ml of chloroform and 250 ml of concentrated sulfuric acid were added. Appearance of the color precipitate indicated the presence of steroids in the sample. Test for phenols. 250 ml of crude extract and 250 ml of 2% ferric chloride solution was added. Appearance of black coloration indicated the presence of phenols in the sample. A test for glycosides. It consists of 2 set tests: Killar-Killarney test and glycoside Liberman's test. 250 ml of crude extract and 250 ml of glacial acetate acid and 1 to 2 drops of 2% ferric chloride reagent in water was added and poured in a test tube containing concentrated sulfuric acid. The presence of brown ring at the interface indicated the presence of glycoside. Second test for glycoside, Liberman's test. 250 ml of crude extract and 250 ml of acetic acid and 250 ml of chloroform was added. The mixture was then cooled and concentrated sulfuric acid was then added. Green color indicated the presence of a glycosteroidal part of glycoside. Test for carbohydrates, proteins and amino acids and gum and mucilages are thereby shown.
So next is the result. The reduction of silver nitrate using the plant leaf extract was viewed by the color change in the reaction solution. The reaction solution of the reduced silver nitrate by leaf extract of Clitoria ternatea as seen in the experimental work. Total ash content. Silica crucible was heated to red hot for 30 minutes and it was allowed to cool in desiccator. About 1 gram of powder sample was weighed accurately and evenly distributed in the crucible. The content was shared over the heater and then burned in a muffled furnace for 2 hours at 600 plus or minus 10 degrees Celsius. The crucible was allowed to cool in desiccator and weighed with the percentage total ash is equal to weight of the ash by weight of the sample into 100. Total ash value of Clitoria Ternatea is 7.5%. The Fourier transform infrared spectrometer. The ethyl acetate extract is Liu C. Ternatea exhibited a characteristic band at 2983.88 cm corresponds to CH. Stretching denote the presence of carbonyl compound, which is in concordance with the who has reported what different compound was identified with the variation in the peak ratio. Antibacterial activity of the AGNPS. AGNP is seen to be alternative antibacterial agent to antibiotic and have the ability to overcome the bacterial resistance against the antibiotic. Therefore, it is necessary to develop AGNPS as antibacterial agent. Among the several promising nanoparticles, AGNP seems to be a potential antibacterial agent due to their large surface to volume ratio and the crystallographic surface structure. The antimicrobial activity of AGNPS against the Escherichia coli, in which E. coli cell treated with AGNPS shows the accumulation of AGNPS in the cell wall and the formation of pits in the bacterial cell wall, eventually leading to cell death. In the same E. coli strain, smaller particle with the larger surface to volume ratio showed a more efficient antibacterial activity than larger particle. Furthermore, the antibacterial activity of AGNPS is not only size but also shape dependent. AGNPS are synthesized by 4 different types of saccharides with an average size of 25 nanometer, showing high antimicrobial and antibacterial activity against gram-positive and gram-negative bacteria, including highly multi-resistant strains such as methicillin-resistant Staphylococcus aureus. As mentioned previously, not only the size is important for determining the efficiency, but also the shape, because AGNPS undergoes shape-dependent interaction with the gram-negative organism E. coli. This is the particle count. Conclusion. A critical need in the field of nanotechnology is the development of the reliable and the eco-friendly process of synthesis of a metallic nanoparticle. Nanoparticles are being viewed as fundamental building block of nanotechnology. Silver nanoparticles play a profound role in the field of biology and medicine due to their attractive physiochemical property. In the present study, we have demonstrated that use of natural, low-cost biological reducing agent and the Clitoria ternatea leaves extract can produce a metal nanostructure. Through efficient green nanochemistry methodology, avoiding the presence of toxic solvent and waste, the use of Clitoria ternatea has the added advantages that is plant can be used by nanotechnology. Process industry prepared nanoparticle can be used as a bacterial and in wound healing, water purification, and also in the field of medicine due to this application. This method is potentially exciting for the large-scale synthesis of nanoparticles. Several nanoparticles obtained from the Clitoria ternatea have very strong inhibitory action against the Pseudomonas aeruginosa, followed by Staphylococcus aureus, Escherichia coli, and the Streptococcus viridans. This is a reference for the use. Honorable mention to President Dr. Shivajirao S. Zondle, Secretary Dr. Geeta Khare, and the Principal of the College Dr. Nitin C. More, who encourage and give guidance to make efficient use of social media and make such informative video. Thank you. Hope you like this video.